this video, we'll talk about apoptosis. And this topic is for USMLE Step 1. It comes under Pathology. It's a high-yield topic. Watch the video till the end. So key points associated with apoptosis. It's a ATP-dependent programmed cell death. So it has intricate signaling details that program the process of cell death. Second, intrinsic and extrinsic pathways are two different pathways that might lead to the apoptosis, but each of these pathways converge at a caspase cleavage event. We'll talk about these pathways in the later half. Third characteristic is the overall histological appearance of an apoptotic cell. So the eosinophilic cytoplasm and the basophilic nucleus is a characteristics of apoptotic cell. There are other characteristics like pycnosis and karyopexis. So pycnosis means condensation of the genome or the chromatins too much, and it's basically making the nucleus non-functional. And karyopexis means advanced stage of nuclear dissolution. So nucleus gets fragmented into small condensed half and gets scattered all over the place. That is called karyopexis. So overall, there would be cellular breakdown, cell shrinkage, chromatin condensation, membrane blebbing, formation of apoptotic bodies which would be phagocytosed by immune cells. One of the hallmark of apoptosis, it doesn't lead to any inflammatory reactions. All of these blobs are actually taken by immune cells and everything would be like taken care of. No inflammation in the area, which is very different from the necrosis. Anyway, the cell membrane in case of apoptotic cell remains pretty intact, whereas in a necrotic cell, cell membrane integrity is compromised. But in apoptosis, cell membrane composition change, most notably phosphatidyl serine, which is remaining in the inner leaflet of the normal viable cell, moves to the outer side when the cell is undergoing apoptosis, and that's a hallmark feature to distinguish normal cell versus apoptotic cell. Even if the cell is in early apoptotic stage, it can be distinguished like this. The seventh point is the DNA laddering. So DNA gets fragmented with the action of DNAs. So there would be like 180 to 200 base pair of DNA fragments all over the place. So these are hallmark cellular features of apoptosis. Now let's talk about the pathways of apoptosis. There could be two distinct pathways. One pathway is intrinsic, another is extrinsic. Intrinsic pathway is basically involving mitochondria, whereas extrinsic pathway does not involve mitochondria. Extrinsic pathway involves specific receptor ligand interaction, which would be clear in a moment. So for example, there are TNF receptors, FAS and FAS ligands which can mediate extrinsic pathway. This triggers the initiator caspase activation. Caspase are protein which are getting cleaved to form an active caspase. Now this initiator caspase does not do the job. Its job is to further cleave and activate downstream executor caspase. So executor caspase is caspase 3. This information is really important. So many caspase can start the cascade, but the nail in the coffin is the cleaved caspase 3. This is the executor caspase. Inactive cleaved caspase 3 get cleaved into cleaved caspase 3, and eventually caspase can degrade the DNA, fragment the nucleus and many other things. It can activate plethora of other enzymes and proteases which can kill the cell. Okay, the intrinsic pathway starts with sensing some kind of damage. So the intrinsic pathway can be triggered by, let's say, irreversible DNA damage, or let's say UV mediated damage. Anyway, there are BCL family sensors which can sense this damage, and there are effectors such as BAX and BAD which can poke holes in the mitochondrial membrane. So when mitochondrial membrane integrity is compromised, cytochrome C is released in the cytoplasm. Generally, it is never expected to be in the cytoplasm. Right now, it's in cytoplasm, and there are specific enzymes such as APAF1 interacts with the cytochrome C. Eventually, with help of other caspases, it ultimately activates the cleaved caspase 3. Cleaved caspase 3 lead to all of the DNA cleavage and breakdown of many other cellular enzymes. Anyway, BCL2, BCL cells, these are other BCL2 family proteins which can prevent the Bax activity and thereby promoting cell proliferation. So it's an anti-apoptotic factor. By the way, after the cleavage of DNA, there are blebs which starts moving out of the cell. This might have fragmented organelles, it might have fragmented genome, and eventually these apoptotic blebs would be taken up by immune cells like this macrophage in this example, and this ensures no inflammation happens. Okay, so apoptosis can be involved in pathology or even it can be physiological. So during development, apoptosis work like a molecular sculpture artist. Apoptosis occurs during the digit individualization in our hand during our developmental process. So it's a good thing. It also occurs when you have like some kind of like a proliferation cue. When it is removed, apoptosis can occur. An example can be drawn from the immune system. The effector T cells can fight pathogens, and let's say their job is done, they eliminated the pathogen. Now there is no point of keeping these effector cells specific to that one particular pathogen, and IL-2 is that trigger factor. When IL-2 level goes down, these cells are actually eliminated by apoptosis. Now there could be other trigger for apoptosis like trauma, radiation, or toxin. All of these can lead to apoptosis, and these are obnoxious or injurious stimuli. So it is kind of like pathology associated as well. Okay, there could be symptoms which are associated with apoptosis or problems in apoptosis. Apoptosis is damn important, and when it doesn't happen, because in one case which is known as autoimmune lymphoproliferative syndrome, where the FAS-L and FAS interaction is abrogated, and as a result the clonal deletion of specific reactive lymphocytes, this process is failed. So there are too many self-reacting lymphocytes, which results in lymphadenopathy and many autoimmune related complications. That means elimination of several cell types via apoptosis is super important for normal physiology. I hope this was informative. If you like this video, give it a quick thumbs up. You can follow us on Instagram, Facebook, and Twitter. You can get many notes displayed like this in our Instagram and Facebook page. Support our channel using super thanks. You can pay via Paytm, PayPal, and UPI. See you in next video.